Good afternoon, everyone. I hope you can all hear me, and welcome to this seminar today. The Biomedical Society and Portland Press are pleased to welcome you to this webinar, which is part of the Biochemistry Focus webinar series. And the topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support early career development. Each webinar will give you the opportunity to ask questions, and then you have to pop them in the question tab. And we also welcome suggestions for future topics and speakers to feature in the seminar series, so please check the website for details. And my name is Espe, and I'm one of the immunologists at the Center for Inflammation Biology and Cancer Immunology in King's College London. And today's seminar is entitled Exploring Immune Responses to Tumors Using Systems Biology Approaches. Our invited speaker will be presenting an overview of his lab research into T cells and anti-tumor activity. But before I hand over to our speaker, I would like to reiterate that questions will be asked at the end of the seminar, so you can write them anytime you want, but we'll discuss at the end of the seminar. And if you have a question, just please type it in the question box as shown in the image on the screen. And we will try to answer as many as we can in the discussion at the end. We have 10 minutes allocated for discussion at the end. So it is my absolutely great pleasure to introduce you all to our speaker today, which is Professor Roger Geiger from the Institute for Research in Biomedicine in Switzerland. So just a really quickly fast run over his career. He did his master's and PhD degree in ETH in Zurich, and during his PhD studies with R.A. Hellenius, he investigated how non-enveloped viruses penetrate the host cell membrane. And then he joined the laboratory of Professor Lanvecchia at the IRB to study the metabolic regulation of T cell responses. And then in 2016, he joined the research group of Matthias Mann in the Max Planck Institute of Biochemistry in Munich, where he received training in mass spectrometry-based proteomics. And from 2017, Roger has lead his own research group at the IRB, where he focuses on immune responses to tumor using biology approaches. So I'm going to tell you that Roger's lab has multiple interests. I'm not very sure he will be able to cover them all, but he has worked from things like quantifying how naive cells respond to antigens, which seems like a naive question to ask, but it's actually really difficult to answer, to understand, for example, things like why T cells fail to control tumor growth. And here he applies very, very interesting approaches and out-of-the-box thinking, which is what I love from his work, to overcome or to propose novel mechanisms on how to overcome the immunosuppressive environment in the tumors by targeting, for example, metabolism or exhaustion. So, Roger, we are delighted to have you here. Thank you very much for accepting the invitation, and the room is totally yours.
Thanks very much, Esperanza, for the invitation. It's a pleasure to be here and present some of our work to you. So can you hear me still, Esperanza?
We can hear you and see your presentation very well.
Perfect, great. So then I go on, and I'd like to start by showing this movie, which depicts a naive T cell that we did based on electron microscopy images. And in purple, you can see the surface of the naive T cell. In green, the surface of the nucleus, and the blue structures are mitochondria that typically sit together on one side of the cell. And so the T cells is the subject that we are focusing on in our lab. Now, when we isolate naive T cells from the blood of a healthy individual and then activate those T cells, then after about 40 hours, they undergo a substantial activation program, grow in size, and then start to proliferate and assist in the clearance of an antigen. Now, the human body contains approximately 200 billion naive T cells that circulate for years through our body, and most of them have a different T cell receptor. And these naive T cells are only ever needed if the body is exposed to an antigen that they recognize through their T cell receptor. So most of the naive T cells are never used, and therefore it makes sense to keep these cells in a very small state that consumes as little nutrients and energy as possible. But then when they, or if they recognize an antigen, these naive T cells have to execute this activation program rapidly, and so therefore they need to be in a prepared state to act as quickly as possible. So there is a trade-off here between energy saving and being prepared to react promptly. And so we are interested in this trade-off and decided to study the organization of naive T cells. And so we asked what kind of proteins are synthesized in these quiescent cells and how rapidly are they turned over. And so we used a mass spectrometry-based approach called pulsed SciLAC that was developed by my former supervisor, Matthias Mann. So we isolate and fact purify naive CD4 T cells from human blood, and then we culture them without a stimulus, just culture them in a medium containing heavily labeled amino acids, here shown in yellow. And these amino acids are then incorporated into newly synthesized proteins, and we can then distinguish between newly synthesized proteins and pre-existing proteins based on a mass shift. And if we do this for increasing times, we can essentially calculate the protein synthesis rate of every protein in the cell. And we know that when we isolate a naive T cell and culture it without a stimulus, that the mass, the protein mass, remains constant because these cells are quiescent. They do not grow, they do not proliferate, they just sit there. So the mass remains constant. So that means for every protein that is synthesized in these cells, another protein has to be degraded, otherwise the mass would increase. And so therefore we assume that what we can measure, protein synthesis, is also an approximation for degradation or for protein turnover. And so that's the assumption here. And so then based on this, we ranked the proteins according to turnover. And you can see that so turnover of 1 means that the protein is entirely renewed within 24 hours. Now, the first observation here was that not all the proteins are renewed in naive T cells. Actually, 60–70% of the proteins are not detectably renewed in our assay, meaning that they are stable, they just sit there for 24 hours stably. And then there is a handful of proteins shown on the left in red, which are rapidly renewed. And here are some examples. Beta-2 microglobulin, for example, this is a subunit of MHC class 1, is renewed within only 45 minutes. So there's 500,000 MHC class 1 proteins on the surface of a naive T cell, and those are renewed very, very quickly, probably to ensure that intracellular antigens are continuously displayed. So here are some other examples. ETS1 transcription factor, HLA, the other subunit, and so on and so forth. I don't know whether you can see my, so I try this here, laser pointer. So here you can see GAPDH. This is a glycolytic enzyme, has a very slow turnover rate, and histones are renewed in 2,000 hours. So practically not renewed, which is consistent with the very compact heterochromatin in naive T cells. And so if you're interested in your protein, how quickly your protein is renewed in T cells, so these data are all accessible through our platform immunomics.ch. And we noted that there is quite a large number of transcription factors that are rapidly renewed in naive T cells. Rapidly means within hours completely renewed. And so we were speculating, why are these transcription factors so quickly renewed? And so one explanation could be, okay, they are renewed to ensure that they're in the correct conformation, in the correct state, to ensure transcriptional fidelity, because if they're misfolding somehow, then inappropriate translation could happen. So that's one hypothesis. The other hypothesis is if these transcription factors are rapidly renewed all the time and need to be down-regulated following activation, the T cell can simply stop their synthesis, and then the protein will automatically disappear without the need of a mechanism through, for example, ubiquitination and so on and so forth, E3 ligase, to degrade them. So it can. So if you need to downregulate the transcription factor, it stops translating them, and then they disappear. So this there's some evidence for this. For example, KLF2 is one of the quickest renewed transcription factor. We can detect it in naive T cells, but then 6 hours following activation, the transcription factor already disappeared. When we look more globally here in T cells that enter tumors and become activated, you can see on the left side several factors that are down-regulated following activation. And so when we then look at their renewal rate here, color-coded, you can see that several of these factors that are down-regulated following activation are of high renewal in naive T cells, so suggesting that this is a potential mechanism to ensure a quick down-regulation of a protein. So to summarize this, the majority of the proteome of naive T cells is stable, but a small set of proteins is rapidly renewed, and those proteins have important functions. So we then asked how many proteins are synthesized on an absolute scale, and we used a relatively old method that was developed by Jacek Wisniewski in the lab of Matthias Mann, and so it looks at the proportion of the mass spec signal of an individual protein to the total mass spec signal, and this proportion is then related to the protein mass. It's a very simple way to estimate protein copy numbers. The protein mass can, of course, be determined by a Bradford assay, and here we have the spectra. So based on this, we estimated that a naive T cell synthesizes approximately 60,000 proteins per minute, which is substantial. And then after activation, this increases to 750,000 or even 1 million proteins per minute. So the next question we asked was, how many ribosomes are then in a naive T cell? And for this, we quantify proteins and RNA, ribosomal RNA, and estimated copy number independently. And so the two data sets showed that naive T cells contain approximately 400,000 ribosomes. And then, of course, upon activation, the number of ribosomes increases. But if we look here in naive T cells, there are about 400,000 ribosomes per cell. And so now we also looked closer in collaboration with Matthias Peter at ETH Zurich into these ribosomes in naive T cells, and we found that they are assembled, they're pre-assembled, but somehow inactive. So when we calculate now based on the data, on our mass spec data, the activity of these ribosomes, so we know the number of ribosomes, we know how many proteins are synthesized per minute, and we know the amino acid sequence of all these proteins, so we can calculate the output in terms of amino acids per second and ribosome. And so the baseline output in naive T cells is about 0.8 amino acids per second. That's, you know, the average across all ribosomes. Could be that some are active and some are inactive, but on average, the output is 0.8 per second. Now, when we activate the T cells using antibodies to CD3 and CD28, then you can see that the output per ribosome almost instantaneously increases to 4 amino acids per second and ribosome, which is, by the way, the maximal output of ribosomes, which has also been determined in cancer cells using different methods. So from this, we conclude that naive T cells contain large numbers of mostly idling ribosomes that can be rapidly engaged following activation of T cells in order to ramp up this activation program. And so these pre-existing ribosomes is what we call a mechanism of preparedness to promptly respond to an antigen. So we then ask, well, is this regulated through the synthesis of messenger RNA? So we could imagine that there are many, many ribosomes sitting there just idling, and then following activation of T cells, the messenger RNAs are produced and can then be read off or translated by the ribosomes. So for this, we quantified the messenger RNAs in naive T cells and also developed algorithms to estimate copy numbers here on the y-axis. So some of these messenger RNAs are present in 0 copies messenger RNAs, so they're not, they sit in, they are present in naive T cells, but they're not translated. For example, there are about 70 copies of mRNA encoding CD69, which is an early activation marker, but of course in naive T cells, CD69 is not translated. And then there is here on the right side beta-2 microglobulin, ubiquitin. Those are highly abundant messenger RNAs that are also translated by ribosomes. And then here are ribosomal proteins that occur in large messenger RNA copy numbers, but are also largely repressed, meaning not translated. So to further illustrate this, we calculated the transcript translation rate, meaning how many times the individual messenger RNA is translated by ribosomes. If you look here, for example, beta-2 microglobulin messenger RNAs are on average read off 5 times per minute by ribosomes. So those are polysomes that move across the transcripts. But here CD69, CD40 ligand, ribosomal messenger RNAs are completely repressed in naive T cells, so not translated. When we then activate naive T cells, all these points move to the right, and now the messenger RNAs are translated, and after 24 hours, there is a good correlation between the abundance of a messenger RNA and the actual protein synthesis rate. Now, from this, we concluded that naive T cells not only contain pre-existing ribosomes, but they also contain pre-existing messenger RNAs that are repressed and then can be translated following activation. Now, finally, we looked into glycolysis energy. So this activation program requires a lot of energy, and we know that the glycolytic rate is increased 5 to 10-fold within 12 hours. And we also know that the glycolytic pathway is rather complex. So we were wondering, how is it possible that naive T cells can upregulate this complex pathway 5 to 10-fold in a short period of time? And so we looked into, now here this is protein abundance, copy numbers on the x-axis here in naive T cells, and copy numbers in 12 hours activated T cells. So by the way, after 12 hours, the T cells still look like this. So the blasting occurs a bit later, around 24 to 40 hours. So here the cells are small. We compare copy numbers, and you can see here the glycolytic enzymes are highlighted in yellow, and they all lie on this diagonal axis, meaning that their relative abundance doesn't change following activation, despite the fact that the glycolytic rate is increased 10-fold. With the exception of hexokinase 2, which is upregulated. It's the only protein that is significantly upregulated. But then we noted here on the upper right corner that there are several glycolytic enzymes that are highly abundant already in naive T cells. And by highly abundant, I mean abundance like actin, tubulin, millions of copies. So GAPDH, for example, is present in about 3 million copies per naive T cell. And so when we then inspect the glycolytic pathway, you can see here for every protein highlighted in yellow, there is a enzyme present in naive T cells in more than 1 million copies. And so from this, we concluded that T cells also, there is some metabolic preparedness, meaning that naive T cells have large pools of glycolytic enzymes that are not needed and are only engaged following activation. What we also know is that these glycolytic enzymes in naive T cells, they are stable, and then following activation, the amount of the glycolytic enzymes doesn't change, but they start to be turned over. So probably they are kind of stored in a superstructure in T cells that then can be engaged following activation. And we're currently studying how these glycolytic enzymes are stored in naive T cells. So to conclude, T cells, naive T cells, have a very thin layer of cytoplasm, and 11% of those proteins are glycolytic enzymes, despite the fact that naive T cells hardly perform any glycolysis. So this is really to be prepared for the activation process. So with that, I would like to move to the next topic, which is more focused on metabolism. As you know, cellular metabolism is intertwined with the functions of a cell, and so therefore we decided many years ago to study metabolism of T cells. And what we found at the time was that the amino acid L-arginine is not only a building block of proteins, but L-arginine is an immunomodulatory amino acid. And when we increase L-arginine concentrations in T cells, then the effector functions and the anti-tumor activity of these cells is increased. And so when I started my lab after this postdoctoral work, I asked whether we can translate this finding to the clinic. And what we did is we used mouse tumor models, MC38, and supplemented arginine, lots of arginine, by oral gavage to the mice, and analyzed whether this synergizes with the anti-tumor response. And what we found was the following. So these are the controls. If mice receive immunotherapy, anti-PD-L1 blockers, then the mice survive longer. But if we, in addition, provide L-arginine through the diet, then about 75% of the tumors were rejected and the mice survived. And so this was interesting, but the problem was that if we wanted to translate this to the clinic, a patient would really need to ingest a lot of arginine. So we were thinking for a long time, how can we locally in tumors increase arginine concentrations? And then we came across E. coli Nissl, which is a non-pathogenic probiotic bacterium that is anaerobic and grows in tumors. It grows inside the necrotic hypoxic core of tumors, where it's not controlled by the immune system. So this is the niche where it grows, like on an agar plate. And we now know that Nissl can be safely administered intratumorally to patients. So our goal was to engineer Nissl such that it produces arginine, which contains a lot of nitrogen. So we decided to use ammonia as a nitrogen source. And it turned out that ammonia is a metabolic waste product that accumulates in tumors up to millimolar ranges. So about 5 millimolar ammonia is present in tumors. So this is perfect to use and convert it to L-arginine that is then enhancing the anti-tumor immune responses. By the way, ammonia also inhibits some parts of the immune system. So by converting ammonia into arginine, we not only enhance T cell function, but there's also other benefits to that. So it turned out that it was known since many years that bacteria can convert ammonia into L-arginine through a multi-step enzymatic pathway. And I like that the microbiologists termed these enzymes ARCH-A, B, C, D, E, F, so I can easily remember them. And the first enzyme in this pathway is ARCH-A. And when there is sufficient amounts of L-arginine present for the bacteria, then it inhibits this first enzyme and the pathway is shut off because it's energy consuming. And so the first engineering step was to replace ARCH-A with a feedback-resistant mutant, such that there is no longer this negative feedback, and the bacteria continuously produce L-arginine. So by the way, this feedback-resistant mutant was also discovered about 40 years ago. So this is old school microbiology. And now, as for every pathway that is important, this pathway is also regulated at the second level. There is a so-called arginine operon, and all these genes are only expressed when the arginine repressor is not there. So if there is a sufficient amount of arginine, then arginine binds to the Arg repressor, and the synthesis of these genes is inhibited. So then the enzymes are not present. So here the obvious engineering step was to deplete this repressor such that the pathways or all these enzymes are constitutively synthesized and expressed. And so when we did that, we found that all the enzymes, so by proteomics, all the enzymes are massively upregulated and the entire pathway is there. So then when we took these bacteria and cultured them in a medium where the only nitrogen source is ammonia, we could see that they rapidly produce L-arginine, while here control bacteria do not produce arginine. So then we had these little arginine producing factories, engineered nasal strain, that we injected intratumorally into tumors of mice, such that they continuously release arginine. So by the way, if we inject bacteria 1 million bacteria, the next day you have 10 to the 9 bacteria in tumors, and then they colonize tumors long term. So they do not go away. They only go away when tumors are eradicated by the immune system. But bacteria colonize tumors long term. What we found was then that this synergizes strongly with checkpoint inhibition. So this is the benefit here of, so this is bacteria, normal bacteria, they have no effect. So this is just like anti-PD-L1, but if the bacteria secrete arginine, the survival rate increases from 40% to about 75–80%. And as compared to daily administration of large amounts of arginine, these bacteria can be injected once or twice, and then they colonize tumors and continuously release arginine. And so now we consider these. So I'm intrigued by these bacteria and using them as a general vehicle, to deliver any kind of payload to tumors. And so we teamed up with Jesse Zalatin from the University of Washington, who is a bacterial engineer. And now we're generating additional strains that not only produce arginine, but also cytokines. And so our goal is then to make multifunctional bacteria. So this is a Swiss knife. I'm Swiss, so I like this analogy. So one project that we are currently working on is on bacteria that secrete interleukin-12. Interleukin-12 is a very potent cytokine, and people have tried to inject interleukin-12 intravenously, but this is very toxic and the patient died. So the goal is to specifically deliver interleukin-12 to the tumors, and we think the bacteria would be ideal because the bacteria selectively grow in tumors, and then one could trigger IL-12 release selectively in tumors. So this is the theory. Just to remind you, interleukin-12 is a heterodimer, and it's linked through a disulfide bond. But when we express it in bacteria, it aggregates, and it's certainly not secreted as a functional protein. So we then used in silico design, because Jesse Zalatin is from the University of Washington. He's the neighbor of David Baker, who recently received the Nobel Prize in protein engineering and protein design. And so based on their expertise, we generated, so this is mostly spearheaded by Jesse, interleukin-12 redesign that is able to fold in bacteria and does not make the bacteria sick. So this interleukin-12 makes the bacteria very sick, so they don't grow. And so what they managed to do is to redesign this protein such that the bacteria are happy secrete interleukin-12, the redesign, and this redesign is functional almost like the real interleukin-12. And so now we're testing these bacteria in vivo. We do not know yet whether they have anti-tumor activity, but certainly so far it has biological activity. So then a few comments on clinical applications. So I mentioned that E. coli can be administered safely into tumors. There was a clinical trial many years ago. There was a study led by Steve Rosenberg in 2002 with other bacteria that were LPS deficient, and those bacteria were actually injected intravenously, and that was tolerated by the patients. The only problem was that the bacteria did not efficiently colonize tumors. So only in 1 out of 25 patients, bacteria colonized tumors. And the problem is so that if we inject bacteria intravenously, they're cleared within 45 minutes by the complement system and by the innate immune system. So also, for example, when we brush our teeth, bacteria get into the bloodstream and they're cleared within an hour. And so that's probably one of the reasons why these bacteria could not colonize tumors. Also, when we work with animal models and we inject a cancer cell line subcutaneously, these MC38 tumors, they grow very quickly in 1 week, and the vasculature is very leaky, and the bacteria easily get into tumors when we inject them intravenously. But in slow-growing real tumors, the vasculature is more intact. Bacteria don't make it easy to the tumors because they're clear, so it's more challenging. And so Jesse Zellatan and I wrote a review on how we could potentially increase the colonization of tumors upon intravenous administration. So one idea is to enhance the vascular permeability in tumors. And another idea that is now pursued by many research groups in this field is to shield the bacteria temporarily from the immune system such that they have a little bit a longer half-life in the circulation and can then find the way to tumors. So once they extravasate into tumors, I don't know, 1, 2, 3 particles, then if they reach this necrotic core, they can grow like on an agar plate, and it doesn't take long until the tumor is fully colonized. So to summarize this part, bacteria are perhaps interesting living synthetic biomedicines. It's a vehicle to modulate the tumor microenvironment for therapy. One can imagine that growing such therapeutic bacteria is cost effective. It's literally growing them in a medium, and currently we have to improve safety by containment, etc. So there's a lot of work to do. But my understanding is that currently, I think 60 clinical trials with bacteria, or in total 60 clinical trials with bacteria have already been done. So this is a new field or something that is emerging. It's not entirely new. So people have started many years ago, but I think it got some momentum now. So, so much to bacteria. I would like now to come to a third topic in the next 10 minutes or so. And to introduce this topic, I would like to show you a movie. And in this movie, you can see here a cancer cell spheroid. And these little dots here are T cells. And so now when I play the movie, if it works, I don't know. I'll try again. Yes, so if I play the movie, you can see some blabbing here. And then the T cells start to proliferate and kill the tumor like a swarm of piranhas. So when you watch this movie several times, so at least I was impressed by the power of the immune system. I think it really illustrates how T cells are very efficient at growing and at killing tumors. And so this made me understand why there are so many checkpoints in place to keep these T cells in check. And of course, there are many ways how T cells can be inhibited. And in a project about liver tumors, we wanted to identify additional mechanisms how T cells are inhibited in tumors or in general. And so for this, we used resected tumor tissues surrounding matched liver tissues and blood from HCC patients. Then we isolated different immune cells, FACS purified them, and analyzed them by mass spectrometry-based proteomics. But before we analyzed the T cells by proteomics, we phenotyped them by flow cytometry. And so this is here from 50 HCC patients, CD4 and CD8 T cells, blood, liver, tumor. And so this shows PD-1 expression on T cells. And so blue means there's not much, and red means there is a lot, so a high percentage of T cells express PD-1. And you can see here there's only a subset of patients that have large amounts of PD-1 on their CD8 T cells. And so these patients, most of them have a viral etiology, HPV or an underlying HPV or HCV infection. And so all these T cells, they are isolated from tumors through the same procedure. And then we ask, okay, what is different between these T cells and these T cells that have low levels of PD-1? And then we went into our mouse spec data, and also these are some FACS plots, and performed a differential abundance analysis between T cells that have high levels of PD-1 according to the FACS analysis, and T cells from patients where T cells had low levels of PD-1. And so this differential abundance analysis showed here that in the proteomics data, we also find a lot of PD-1 on those cells, which confirms the flow cytometers. That's a good sign. And then we identified this signature here of proteins that are upregulated on PD-1 high cells. And so these are the 22 proteins upregulated. And so we asked, well, are these just upregulated due to the canonical T cell activation program, or is there something special about these proteins that only occurs in tumors? And so for this, we took CD8 T cells from healthy donors, activated them with anti-CD3, CD28, and then expanded them. And so this is the canonical activation program. And you can see that 19 out of these 22, so these are 22, and 19 of those are activated during the canonical T cell activation program. So there's nothing special about those. It's just upregulated in any kind of T cell activation setting. But 3, TOX, AFAP1L2, and LMCD1 were not. So there's something special or tumor specific about these proteins. So then we said, okay, T cells in tumors are continuously stimulated, and we performed this chronic in vitro stimulation assay. So here in blue, this is a canonical activation. So T cells are activated for 2 days and then just cultured. And you can see that PD-1 is not high, and so these are markers of exhaustion. But if you focus here on the chronic stimulation, where we activate T cells for 14 days, you can see that these markers, TIM3, CD39, and LAG3, are upregulated strongly, meaning that we can sort of get exhausted-like cells in vitro. And so this is an assay that many labs use. So now under these conditions, we found that at least one of these 3 proteins, AFAP1L2, was strongly upregulated. And when we estimated copy numbers, you can see here that during the normal activation of T cells, AFAP1L2 is not there. It's only upregulated upon restimulation, strongly upregulated upon chronic stimulation. And so AFAP1L2 is not only present in chronically activated T cells, and according to our proteome atlas of human immune cells, it's also expressed in activated NK cells. So then the last experiment that we did is we knocked out AFAP1L2 in murine T cells, and we found that those cells that are lacking AFAP1L2 control tumors better, indicating that AFAP1L2 is a negative regulator of T cell activity. When T cells in tumors are chronically stimulated, they upregulate AFAP1L2, which inhibits their activity. So it's a negative feedback. If we knock it out, then they work better. So it could be a potential target for immunotherapy. So with that, I conclude. I'd like to acknowledge my group in Bellinzona. Recently, I also started the lab at Technische University of Dresden. So we're setting up this lab and move part of it over there. And I'd like to thank you very much for your attention.
Fantastic. Roger, thank you so much for keeping it to time. Now, just to remind everybody, a lot of you have been very good and already write some questions in the questions tab, but I'm sure there's a lot more coming up. So we're just going to start our discussion. But see, as I told you, Robert has shown us really beautifully how you troubleshoot as you go along when you have a really clear, really interesting scientific question, and you know his imagination and his ability to approach a problem is just mind-blowing. So, Roger, are you happy to take any of the questions? Some of them are well, all of them are really interesting, but I'm going to start from a very kind of target one from Samantha Gibbons. So basically, and I'm curious too about this, she asked how long did it take the T cells to destroy the tumor cells in the video that you show us? How long was the time lapse?
4 days.
There you go, Samantha. That's the answer to your question. There's a lot of questions about the methodology that you use. Again, coming back to Samantha, that she just wanted to know exactly how you kind of calculate the rate to between translation and mRNA. And I think that's something that interests us all because there's a lot of transcriptomics data. Proteomics is more difficult, but there's not much of correlation between one and the other. So
the protein translation was calculated by this pulsed Silac approach that I showed. So we, you know, with the heavy amino acids, but then you check how much of the heavy amino acid is incorporated over time. Then we use this algorithm from Jacek Wisniewski to estimate copy numbers. And so then we have a number on the x-axis, and then we just correlate this to the FPKM. So from the FPKM, so this is a relative number, we quantified then the so we quantified the amount of messenger RNA in cells. And because we know the length and everything, we could then calculate how many copies are present. So we just compare the two numbers. Simple.
Okay, that was very good. There is some questions about where the ATP comes from in naive T cells. So you know the textbooks tell us they come from glycolysis, and this is a question, sorry, by Aditya. I hope I pronounced your name correctly. I'm sorry if I don't, but she says that, or he says that the energy in the naive T cells is or the ATP comes from glycolysis, but the abundance of glycolysis enzymes is constant. But there's difference. Is there any other differences in other energetic pathways?
So the energy in naive T cells mainly comes from mitochondria. They're the main producers of ATP under steady state. Also in activated T cells, mitochondria play a role, so they produce a lot of ATP. And then in proliferating cells, glycolysis is also required. You know, it's this anabolism. Glycolysis also produces ATP, so it's used for both. I think that, did I answer the question?
Yeah, I think so. I think maybe the question was more related to within, and it was something that I also wanted to ask. Within your incredible data set, can you actually calculate things like metabolic flux? So knowing abundance of protein, and I know this has been done with transcriptomics data, can you actually interrogate the flux of metabolites? Because obviously your proteins are in a context of a cell. The cellular location of the protein that is not picked up by the Silac might also, you know, establish some differences on whether the function of that protein is, I don't know, a transcription factor or a glycolytic enzyme or something like that. So, and that will change the whole metabolic flux of the system. Is there a way that you can implement that or understand how other metabolic pathways have an impact?
So we can also, based on the abundance of proteins, or so essentially we can look at the abundance and renewal of proteins with proteomics, and then we can also infer or start hypotheses about metabolic flux. If we want to really look into metabolic flux, we have to label metabolites 13C and then do the proper flux. I think there is no way around proper flux metabolomics analysis for these questions.
There's another very interesting question. I'm moving into your tumor model from Dishat Kadir, that is asking, given the heterogeneity of tumor microenvironments, how reliable is systems immunology approaches in predicting which patients will benefit from the interventions like the L-arginine or another probiotics? Or so how can that inform personalized medicine or not?
So how general this is? I mean, it's, for sure, every patient is different, but so we try to identify common themes. The common theme here with the arginine is such that it does not really affect, I mean, it can affect the tumor. I come to that in a second, but what we're doing with the arginine is boost the T cells. Arginine does not enhance the activation of T cells, so it only enhances T cells that are already activated. So, for example, a cold tumor that has no ongoing anti-tumor immune response, and you add their arginine, nothing happens. So we need a hot tumor, combination with checkpoints, so there is an ongoing anti-tumor T cell response, and the T cells, they themselves upregulate arginase 2 to decrease their own arginine levels. And so if, you know, this is a negative feedback also. So when we provide their arginine, it does something. So it's generally good. It's not good for tumors that grow quicker when they get arginine. So this seems to be the case, for example, for liver cancer, where the arginine not only affects T cells, but also the liver cancer itself. So I don't think it's good for every tumor type, but generally it could be a good therapeutic approach.
I mean, I totally agree with that. So I have a question following up a little bit on that, which is about, I was curious to see that the bacteria actually are happy and survive in the tumor once, you know, they get there. When you try in the humans, it didn't work that well. And one of the strategies that you suggest is shielding the bacteria from the immune system. Now, don't know how risky that is. I mean, maybe, I mean, if you, for example, teach a bacteria how to escape complement regulation by making it express, you know, complement inhibition, is that not a bit risky for human health?
Absolutely. No, I mean, that's so the approaches that have been proposed were to add some polysaccharides to the bacteria just to shield them a little bit, and ultimately the immune system can get rid of the bacteria in the periphery. So when, for example, so here's to understand, if we inject the bacteria intravenously into mice, one day later you find the bacteria everywhere in every organ, not in the blood for 1 hour, and then in the organs, in every organ for about 2 days at very low CFUs, 10 to the 4. And then after 3 days, 4 days, you see in the tumors, oh, it goes to 10 to the 9, but it's cleared in all the other organs. And so if you add these polysaccharides, for example, so French group has pioneered this, then you just allow the bacteria a little bit longer to move around in the blood, but ultimately they're also cleared. And so we decided, Jesse and I decided, if we go down this pathway and we try to keep bacteria longer in the circulation, it's dangerous. So we first spend a lot of energy in making the strains safe, such that they rely on many factors, such that we can easily kill these bacteria. Because it's also dangerous to experiment with these things. And so we said safety first. We make strains that we can control well if something goes bad. And then we try these methods to keep them longer in the circulation.
Okay, I see. Then, so we've established that you need a hot tumor in order for the bacteria to work.
Yes.
And this is a question of myself, and I'll start reading other people's questions too, but it's just to link a little bit with the AFAB1L2. Do you think your new discovery that might help this, because I mean it is a cytoskeleton protein. So what do you think this protein is doing, and why? Do you think it might be implicated on how the cells migrate in the tumor, go to the tumor, how they are able to perform their cytotoxicity functions, or why is this protein bad?
Yeah, so AFAB1L2 has been associated with T cell migration. We know, so now it's very clear also in CAR T cells that are repetitively stimulated, AFAB1L2 comes up. So I think, so we have shown in that 2023 paper that AFAB1L2 is clearly linked to apoptosis. If you upregulate AFAB1L2, cells die also, the most susceptible. So if we experimentally express AFAB1L2, cells die, T cells. And now we also know that when we knock out AFAB1L2, the killing efficiency is increased. So now what is known, so if you look at the structure of AFAB1L2 based on AlphaFold 3 prediction, it's a big protein with many unfolded regions. So it's a fold that is not defined. So it's a huge scaffolding protein that probably plays a role in many different signaling pathways. So at the moment, even though we have worked now a couple of years on AFAB1L2, we still do not entirely understand what it does, but it seems to play a role in TCR signaling, in regulating apoptosis, perhaps regulating migration. It's only there in CD8 T cells. It's never expressed in CD4 T cells. That's what I can tell. But we don't really understand what it does.
But we have another very interesting question about your first part of the talk from Asra Ataisi, which asks, why can some proteins in resting T cells sit and not renew while others are renewed? What kind of speciality made it possible for the proteins to be so different?
So I think the renewal depends on, so I think degradation is independent from synthesis. So one experiment that we did is we added cyclohexamide, which inhibits translation. And then we saw that if we block synthesis, they just disappear. And so that means that this has to be very tightly, so synthesis and stability has to be very tightly coordinated by evolution. It's not a simple process. And so the half-life of a protein certainly depends on, regions that can be ubiquitinated or whatever, their stability. And my guess is that nature has evolved for every protein, not only the function, but also the abundance and the life cycle. So for example, it makes sense. So P62, which is involved in autophagy, and T cells perform a lot of autophagy when they're in a naive state. So this is rapidly renewed. It's a specific feature of this protein, the renewal. It's just, I can only wave my hands here, but apparently evolution has figured out which protein has to have which half-life and renewal rate. Yeah.
Yeah. Okay. So we're going to take one more question from Oluwaseun Owolabi. Apologies for my pronunciation, but the question is: do you think IL-12 is more likely to succeed as a monotherapy or as a combination? So I'm asking, making the question more general, do you think your bacteria could, the development of engineered bacteria with whatever genes you put inside can overcome the need for immunotherapy?
That's a good question, whether we should use it as a monotherapy or combination with other things. I mean, now, hopefully soon we'll find out the impact of IL-12 itself delivered by bacteria. But maybe we need to combine it also with other things. So at the moment, I can only speculate. I would be, of course, pleased if it would work as a monotherapy, but maybe we need to combine it with checkpoint inhibitors or other cytokines as well.
And again, you need the T cells present, right? Like always, that's always happening on the side. Like the first.
It also acts on other cells. So that's also, there's some hope, you know, that other parts of the immune system could help there.
Okay, very well. So I'll thank everybody for coming here today, and thank you so much for your very, very interesting questions. I really hope you've enjoyed the seminar as much as I did. I just, let me just quickly check my notes, but you can continue always the conversation online, so please follow the Biochemical Society either in any of the channels like X or Blue Sky. We also remind you all that we welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. If you have any idea for a webinar in 2025, please submit a proposal, and you can find more information in the webpage. All of the forthcoming seminars and upcoming seminars are listed there, and if you have missed any of them, and there's over 90, you can watch them online in the YouTube channel, as you can see the details in the screen. The recording from today's webinar will also be available to watch within the next couple of weeks. Finally, I really want to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. So please join the Biomedical Society's community of researchers and specialists and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. So please visit the website to find out more about all the activities of the Society. It is really important, especially at these stages of your career, to network and to learn from everybody. So the Society provides an excellent platform for you to do all this. So without further ado and keeping it pristine to time, I would like to thank Roger very, very much for accepting the invite and giving us this incredible and very inspiring presentation. I'm sure that video of the anti-tumor response will stay with us for a long time. And I also thank you all for sticking with us and asking all the fabulous questions. I'm very sorry I didn't go through them all, but we didn't have enough time to discuss them. And please be in touch with the Biochemical Society for future events. Thank you all very much, and I'll see you somewhere.
Bye-bye.
Bye.